Hi, in this video we're continuing to explore how to do repertoire analysis with Platforma. And in this guide we're going to do a simple time tracking analysis to track the abundance of specific clones across various time points or in general conditions. So let's go to our demo project where we have already clonotyping done, and now downstream we have our clonotypes. Well, the time tracking analysis consists of two parts. The first part is to select which clonotypes we are going to track. And in this case, let's start with a very simple thing. Let's just add a clonotype browser block and use clonotype browser to tag certain clonotypes. So here we're going to focus on the immunoglobulin heavy that we have in our data. And once the data is loaded, we have just a table of all of the clonotypes that we have in our project. If you go to overlap, then here you see that in total we have 1,700,000 of clonotypes across all of the samples that we have in the data. So let's select a group of clonotypes by some criteria. It really depends, and track them across different time points. Remember, in the previous guides, the project that we're considering is immunization campaign. So, for example, in the clonality analysis, we saw that at the 8th day after the first immunization, there is a clonal expansion, a clear signal of clonal expansion. So let's just take top clone types from 8th day after immunization 1 and track them across time points. Let's go to samples and data just to see, like for the 8 days after immunization 1, we have multiple samples. Let's select one of them like this, just for simplicity, and go to the annotation tab in Clonotype browser. Let's call this annotation immunization 1 responders, meaning that those are the clonotypes which are responded to the immunization 1. And now I am adding the annotation that I am going to call top 10, 8 days. Okay, so to filter which clonotypes we are tagging as top 10 after 8 days, we're selecting a column with the fraction of UMIs, this data has UMIs, as our filter criteria, and the predicate is top 10, selecting top 10 clonotypes. Let's run this and go to the annotation stats. So with this annotation, what actually we'll do, we will assign a label top 10 after 8 days to every clonotype that satisfies this criteria. We may add more annotations, we may add complicated and comprehensive filters, etc. But for this simple purpose, that's it for now. So here on the annotation stats, you see the statistics, how many of such clonotypes we have in each of samples from the project, and what the total fraction of those clonotypes calculated in terms of the UMI. So really depending on whether we have UMIs in our bulk data, or if it's single cell, that will be a fraction of cells actually, yeah, in each sample. Also, if we go to the, for example, overlap, then among our columns that we have for the overlap, we have many columns, all of the abundance of all the clonotypes. We have a column called im1 responders, which is the column, well, empty values of this column, meaning that this clonotype was not assigned the tag that we generated, top 10, 8 days. And for some of the clonotypes, if we sort by the thing, then we will see these 10 clonotypes that we have assigned our annotation. And what is interesting actually, that those clonotypes, and that the pattern we see immediately, that all of those clonotypes are actually presented in many of our samples. So it looks like the following. It's just a hypothesis that after the immunization, which is like the second time point in our analysis, like we did immunization, yeah, and then the clonotypes that are expanded here, they started to expand after the second day of immunization. They are present in all of the remaining samples, all of the remaining time point that we have. That's the hypothesis. So let's now look on the time tracking analysis of those specific clonotypes. And to do that, I'm introducing a very powerful block that we have on the platform. This block is called Graph Maker. And this is literally graph maker, a tool allowing to build a comprehensive publication-ready visualizations of many different types, bring statistics, etc. In our previous guides of the repertoire diversity, you already saw how we use this graph maker to play around with the visuals. So now, to build a time tracking analysis, I am adding a stacked bar plus stream area chart. And now let's build our time tracking analysis. So what do we want to show on the y-axis? Well, obviously on the y-axis, we are going to show the fraction of UMIs. For immunoglobulins, heavy chain, that's the clonotypes that we selected. We are selecting immunoglobulins. Remember, in this project, we have all other chains. And now we don't see the graph because for the stack bar plus string area chart, we need to select the primary grouping. And as a primary grouping, we can use different things. Let's use time point. So time point is our primary grouping. So on the x-axis, we now will have time points. And also to, so right now, when we did this, every bar contains all of the clonotypes across all of the samples presenting in this time point. So let's filter to only those clonotypes that are our responders. So in one responders, that's the annotation that we created, and top 10 8 days is the one I'm going to use to filter only those clones that present in the annotation. So already something more interesting. And the final touch here, what do we want to track? Obviously, individual clonotypes. And this can be done in different ways. So one of the ways is to use just a CDR3 amino acid sequence as secondary grouping. So that's what I'm doing here. Yeah. And now we have a beautiful time tracking visualization. It's not yet ready. We will continue. But before that, just to note that we can also use not necessarily the CDR3. We can use, let's say, best gene, just for example, yeah, as a secondary grouping to time track the genes, the individual genes. So all of the clonotypes belonging to those genes, and then here we'll have the genes and the total abundance of the genes. So, or we can use, for example, clone label. If you can notice then that the clonotyping analysis assigns a clone label to every clonotype, and we can use this label here as well. But what we are going to use, we are going to stick to the CDR3 amino acid sequence. And you see with how, with a great easiness, I'm just dropping parameter variables and options from left to the right and how interactively the visualization is changing. So our visual is not yet ready. We need to go to the access settings, to the primary grouping, and to actually, well, sort time points here according to the real timeline, because the human labels that we assign are not necessarily correspond to the real time timeline. So I'm going to sort the data according to the time points, yeah, to the timeline, and yeah, so basically that's it. And also, let's say, for example, here I'm focusing only on the first mutation campaign, so I'm making hidden all other time points, and now we have our time tracking visualization. So here we have a total fraction summed, and that's the option. We can actually average by the samples in each time point, and yeah, that's it basically. What else we can do? This is like already a nice thing. We can further fine tune, and then in other guides we'll have a more comprehensive explanations how to fine tune this stuff. But another thing which I also like here is actually removing the CDR3, and just to see that our clonal dynamics is really correct one. We have a clear clone expansion. I'm using a box plot plus jitter dose to see that every dot right now is an individual clonotype, and we see like clearly that the 5th days and the 8th days, those top 10 clonotypes that we selected top 10 at the 8th day after immunization one, they are dominant, while yeah, they are expanding, and now they are declining in their abundance. So as usual, we can bring statistics on the plot, we can add a p-values, we can do let's say, group comparisons. Well, there are many significant comparisons here or this time points, or we can use a reference group, let's say control before immunization one as a reference to to apply statistic tests for the control and all other points. So that's it. The remaining thing is to actually export this graph. Let's call this graph time top 10, 8 days in days at 8 days time tracking. We can export this plot and just include right away in our publication. One thing to notice here, very important, the way how we selected clonotypes. So we used a very basic approach right now. We just used the annotations in the clonotype browser to annotate top 10 clonotypes for one of the samples that we choose. Actually, there are more comprehensive ways to select the clonotypes, to find the clonotypes. We have blocks for differential abundance analysis. We have blocks for specificity prediction and many things, allowing you to find specific clonotypes and then track them or visualize in different ways. That will be shown in the further guides. Stay tuned and thank you all for watching.